Hello, good afternoon, class. In the I hope I'm audible. Okay, so in today's class, we will be talking about Module 3, which covers your quality of the drug substance and the drug product. In today's class, we will focus on 3.2s part of quality module, which specifically talks about the quality of the drug substance. Okay, so in today's session, we will, learn about the harmonized structure of how we can present the chemistry, manufacturing, and control related information in drug registration applications. Okay, we'll also emphasize on the GMP and risk management in today's class. Apart from that, we will be covering the validation criteria for analytical procedures, including qualitative and quantitative tests for active substances and impurities in the APIs. So, whenever I'm saying API, it's synonym to drug substance and vice versa. So, drug substance is and will be your active pharmaceutical ingredient. Okay, so API is basically drug substance is basically a raw material from which you manufacture your finished product. Okay, so today we will be covering drug substance, and if the time permits, we will move on to the drug product as well. So let's start with module 3, part S, drug substance, also known as 32S of module 3. Okay, so the first section of module 3, 32S, is numbered as 32S1, which is general information. Okay, general information is further subdivided into nomenclature, structure, and general properties. All these headers that we are talking about are directed towards drug substance only. We are not talking about anything that is related to drug product right now. Okay. So nomenclature. In nomenclature, we will have. So this is how a DMF is made. We've covered module 1. We've covered module 2, which was summaries and overview of module 3 to module 5. Okay. And now what what are the main data that goes into into making a DMF or doing a submission for registration or a variation, etc., to the regulatory bodies. So here we will talk specifically about the data that you have to submit in order to get the approvals. So the first thing that you have to submit in 32S is 32S1, which is general information, and it covers the nomenclature, structure, and general properties of the drug substance. Okay, so in nomenclature, we have to provide the chemical abstract service registry number. So now what is CAS registry number? It's a unique numeric identifier assigned by a chemical abstract service to every unique chemical substance described in the scientific literature. So every chemical substance described in the scientific literature has a CAS number, which is provided by the CAS services. Okay, chemical abstract services provide that registry number to every unique chemical substance. So we have to provide the CAS registry number in the nomenclature. Then we have to provide the recommended international non-proprietary name, which is referred to as INN. Every drug substance has an international non-proprietary name along with the chemical name for the drug substance. Okay, so these are three important things that are required in the nomenclature. First is your CAS registry number. Second is your international non-proprietary name, and third is your chemical name. Okay, then is your structure. When we are saying structure, we are talking about the structure of the drug substance or the active pharmaceutical ingredient. Okay, in structure, we need to include, first of all, structural formula of that API, along with the relative and absolute stereochemistry, molecular formula, relative molecular mass, and chirality. All these data should be submitted under structure, which is your 32S12. This general information is your 3 to S1, right? So general information is your 3 to S1. This is your S12. This is your S12. This is your S1. This is your S11. This is your S12, and this is your S13. S13 is general properties. In general properties, a list should be provided of physiochemical properties such as pH, pKa, melting point, solubility, hygroscopicity, physical form, and crystalline form. Okay, apart from this, a list of the polymorphic form present in the proposed active substance should also be included under general properties. Only a list of the polymorphic form present in the active substance. No details about the polymorphic form should be provided in general properties. Only the physiochemical properties of drug substance will be provided in general properties along with the list of polymorphic forms present in the active substance. Okay? Is 3 to S1 general information clear to everyone? You can just show a thumbs up if it's okay. Then we'll move on to 3 to S2. Okay, so moving on to 3 to S2. 3 to S2 majorly talks about your manufacturers. Okay? So this is your 3 to S and 2. Okay. Under 3 to S2, there are six sections. Okay. The first one is your manufacturer's detail. In manufacturer's detail, you have to provide the name, address, and responsibility of each manufacturer, including contractors, all the vendors, and each proposed production site involved in manufacturing and testing. Take for example, there are three manufacturing sites which is involved in the, you know, in the production of the API or the testing of the API or the packaging of the API. So details of all those manufacturing sites along with the responsibility of each manufacturer must be provided in your section 32S 21. Okay. Then comes your 32S 22, which talks about description of manufacturing processes and process control. Here you will have to provide or submit a flow diagram of the synthetic processes and a sequential narrative of the manufacturing process. First thing that you have to submit here is a flow diagram, and second you have to submit is a sequential narrative. Sorry, just a second. Okay. Second, you have to submit is a sequential narrative, right? This will include the flow diagram, and the sequential narrative should include the quantity of the raw material that you're taking, the solvents that are used, catalyst, and reagents, reflecting the representative batch scale for commercial manufacture. Okay. All the process controls will be identified in 32S22, and if there are any critical controls that needs to be addressed, they will be talked about in section 32S24 of manufacturers. Okay. So the first one is manufactured, where you are talking about the manufacturer's details. Second is your manufacturing process and process control. The third one is your control of material. So in control of material, you have to submit information on the quality and control of materials used in the manufacture of drug substance, like raw materials, starting materials, any solvents, reagents, or catalyst should be listed, identifying where each material is used in the process. So all the material that are used that will be used in the production of the drug substance will be mentioned under 32S23 control of materials. Okay, for biological For biologically sourced materials, it will also include information regarding the source from where that is obtained, who is the manufacturer, and what are the characterization of that product. Okay. Then is your 32S24, which is control of critical steps and intermediate, where you have to provide test and acceptance criteria performed at critical steps identified in 32S22 process description. Okay. So, whatever critical steps that you identified here, the test and acceptance criteria should be included in 32S to 4 of control of critical steps and intermediate. Then comes your 32S to 5. Okay, here's the slide share for control of material and critical steps. We've talked about material quality. We've talked about biological materials here and the critical steps, right? So these are what data related to the material quality, biological materials, and critical steps will be provided in 32S to 4. Then comes your process validation and evaluation, which comes under section 32S 32S to 5. Okay, sorry, it's part of your it's part of your manufacturer 32S2, and it's the fifth section. It's just refer to it as 32S5. Okay, 32S25, which is process validation and evaluation. So under process validation and evaluation, it's all the studies for aseptic processing, sterilization should be included in process validation and evaluation. The aseptic process may be recorded through comprehensive documentation. For example, in aseptic processing, you have to include data related to process validation or evaluation studies for aseptic processing and sterilization, and ensure that suitable testing facilities, equipment, instruments, and methodology are available. Because when we are talking about aseptic processing, we need an aseptic environment to carry out the process. So we need to make sure that suitable testing facilities, equipment, instruments, and methodologies are available. Then come your clean room facilities, where you need to ensure that suitable clean room facilities are available in terms of both local and background environments. Local is the environment that you are actively working on, and background is your environment, which is which is the surroundings which might affect your API. So you need to make sure that the local and background are clean, and if not, you need to implement a program of free testing, in process control, and monitoring in order to make sure that the clean room facility is as per the GMP guidelines. Then is your reprocessing, which will include justification for reprocessing as part of the manufacturing process description. Reprocessing, if reprocessing is warranted by a regional authority, then the information for reprocessing will be included in your section 32S22. But if there are critical controls associated with the reprocessing operation, the critical control should be included in 32S24. And if there are validation information which is warranted, it will be included in your section 32S25. Only if there are validation information. Okay. Then we will talk about reprocessing in 32S25. Else, for critical control, it will be covered in 32S24. And if it's warranted by a regional authority, it will be covered in 32S2. Okay, so this is about 32S25 process validation and evaluation. The next part of manufacturer is manufacturing process development. Okay, in manufacturing process development, a description and discussion should be provided of the significant changes made to either the manufacturing process or the manufacturing site of the drug substance. So, if there are any changes in the manufacturing process of the drug substance, or the manufacturing site where the drug substance was initially produced, that needs to be documented in manufacturing process development, and references should be made to drug substance data provided in 32S 44, which we will cover in the next slide. Okay, so any change to the manufacturing process or manufacturing site will be documented, and a discussion will be provided in 32S to 6. Okay, I'll just write 32S to 6 for easier. 32S to 6. Okay, 32S to 6 is your manufacturing process development. Okay, to better remember it, you can just MFD, MPD, manufacturing process development. Okay, so this is about your 32S2, which is your manufacturer's data. Then comes your characterization, which is 32S3. 32S3, which talks about characterization. In characterization, we'll talk about structural elucidation and other characteristics along with different types of impurities. I hope everything's clear till 32S2 manufacturers. If there are any questions, you can write them down in the chat box. And then we'll move to 32S3, characterization and impurities. It's very important for you to understand what kind of data is being submitted. So if there are any questions, please drop them in the chat box and we can take them up in the next class or by the end of this class. So coming back to characterization, the first thing that we talk about is structure elucidation and other characteristics. So in structural elucidation, you have to provide confirmation of structure based on the synthetic route and spectral analysis. This is all part of your chemistry manufacturing control, where you are confirming a structure of the API based on the synthetic route and spectral analysis. Okay, it will include information such as potential for isomerism. If the API, if the drug substance has the potential for isomerism, or identification of stereochemistry and potential for forming polymorphs. This is very important because without this, you will not understand the polymorphs that are being formed during the processing of the API. And if you have an understanding of the different type of polymorphs that can be formed during the processing or the manufacturing of the API, we can always, include critical controls to make sure that the polymorph is not acting as an impurity. And if it is acting as an impurity, what are the steps that can be taken to, remove that impurity, to identify if the impurity is hampering the drug substance quality, or is it okay to have a certain limit of that impurity in the drug substance. Okay, so we need to understand what kind of potential the drug substance has for isomerism and for forming polymorphs. Okay, so under structural elucidation, I hope you understand what kind of information needs to be submitted: the isomerism, stereochemistry, and polymorphs. Then is your impurities. So, when we talk about impurities, we need to provide information on impurities, including classification and identification of the impurity, report generation, listing of impurities in specifications, and a brief discussion of the analytical process. Okay. For example, organic impurities. So, you need to address organic impurities. Uh, Both process and drug related. Other impurities like inorganic impurities, if there are any residual solvents, if there are any genotoxic impurities. See, impurities is again a different topic to be discussed. So I'll just brief you here about the types of impurities and what needs to be addressed under impurities. Okay. If you refer to, ICH-Q3A guidelines, it will ICH-Q3A guidelines, it will tell you about the chromatograms that are provided as part of analytical validation studies related to the impurities. So there are different segments for impurities which are talked about in different ICH guidelines. I'll share you the ICH guideline for reference towards the end of this class. So yes, under characterization and impurities, we have to submit elucidation of structure and characteristics along with the type of impurities, their classification, report generation, specifications, and a discussion on analytical procedures, right? This is 32S3, which is divided in two sections. Now we will talk about control of drug substance, which is 32S4. Okay, control of drug substances and specification. So 32S4 is further divided into five sections, which covers specification, analytical procedure, validation of analytical procedures, batch analysis, and justification of the specification. Why that specification has been selected. Okay. So under specification, you have to define specifications as a list of test references to analytical procedure and appropriate acceptance criteria, which are numerical limits, range, or other criteria for the test described. Take for example, you are submitting a specification. There are different tests. Take, for example, dissolution test. So, a specification, a range is being predetermined, and that specification is provided here that yes, the drug substance should fall under this specification. If it's above and beyond or below, what action should be taken, or if the drug substance is or if the quality of the drug substance is suitable to manufacture the drug product, right? So we need to make sure that a list of test references to analytical procedure and appropriate acceptance criteria is provided to test the quality of the drug substance. Okay. We can also provide a copy of the monograph, including description, identification, assays, and impurities under specification. The next is your analytical procedure. The under analytical procedure, we have to provide the analytical procedure used for testing the drug substance, including identification test, qualitative test for impurities, limit test for control of impurities, and quantitative test of the active moiety. See, all these terminologies will be better understood by you if you are working in a laboratory before moving to regulatory. So if you have experience in chemistry labs or in manufacturing plant, you will be able to understand what these qualitative tests are, what are the limit test for the control of impurities. So yes, all these information related to the drug substance is submitted under analytical procedures. Okay. Then comes your validation of that analytical procedure. You cannot just go on and perform an analytical procedure. You need to validate the analytical procedure before performing validation. Okay. So, a description, analytical validation information, including experimental data for the analytical procedure used for testing the drug substance, should be provided here in validation of analytical procedure. Other two parts of 32S4 are the batch analysis and justification of specification. In batch analysis, description of the batches and result of batch analysis should be provided. All residual solvents should be removed to the extent possible to meet the product specification, good manufacturing practices, and other quality-based requirements. So, all of the residual solvent needs to be evaporated or removed from the final batch that will be released. Okay. Then is your justification of specification, under which Justification for the drug substance specification should be provided. For example, a summary of data from other sections with a cross reference to the detailed information can be provided to support the justification of specification. That why was this specific specification was selected for the drug product. You can cross refer to all the data that you've submitted and provide a justification here in 32S 4 5. Okay, so this was about control of drug substance and specification. It's divided into 5 parts, and it mainly talks about specification, analytical procedures, validation, batch analysis, and justification of specification. Then comes your 32S 5, which is reference standards or materials. So a reference standard or a reference material is a substance prepared to use as the standard in an assay, identification, or purity test. For example, when we do a when we do testing of products using a UV spectrophotometer, what happens using UV spectrometry? We use a reference material and test the sample against it. Just like that, here we are using a reference standard or reference material as the standard in an assay identification or purity test against the API. Okay, we use a pure API and we submit the API that we are manufacturing to test if it's meeting the quality requirements. Okay, all analytical results of reference standards or reference materials used as a reference substance will be submitted under 32S 5. Okay, so this was fifth part of your drug substance 32S, right? Then comes your 32S 6, which talks about container and closure systems. I will share with you the just a second. See, so here it's given 32S 5 reference standards or material, where when you will see 32S 5, information on reference standards or reference material used for testing of the drug substance should be provided, and the ICH guideline to be referred is Q6A and Q6B. Then 32S 6, which talks about the container closure system for drug substance. Here you have to submit a description of the container closure system, the identity of the material of construction of each, the primary packaging in which the product is being stored, sorry, drug substance is being stored, and their specifications. The specification should further include description and identification, any critical dimensions with drawing where appropriate, and along with non-compendial methods should be included where appropriate. Okay, so 32S 6 majorly talks about your container closure system for the drug substance. For non-functional secondary packaging, those that do not provide additional protection, only a brief description should be provided. For functional secondary packaging component, additional information should be provided. The suitability should be discussed with respect to choice of material, protection from moisture and light. What's the point of container closure? The containers are being prepared to, protect the drug substance from moisture, light. We need to also check the compatibility of the material of construction with the drug substance. We need to make sure that the drug substance is does not come in contact with the uh, container or the material of the container and undergoes a reaction which is impurity or a substance that does not have medicinal property. So we need to make sure that the material of construction is compatible with the drug substance, including sorption to the container and leaching or safety of the materials of construction. Right, there are a lot of parameters that needs to be considered while constructing a container closure system for the drug substance. The last section of 32S is 32S 7, which talks about stability. Under 32S 7, there is stability summary and conclusion, post-approval stability protocol and stability commitment, and lastly is your stability data. For stability, you can refer to Q1A, Q1B, and Q5C. ICH guidelines. Moving back to our slide share, I'll discuss the stability there. Okay, so this was control of drug substance, stability, and container closure system. I have summarized a lot of things in this slide, but for details on stability and container closure, you can always refer to ICH guidelines. Okay, so in stability, the first one that we talked about it, summary of stability summary, where we have to provide the summary of all the stability data that we've gathered under 32S73, which talks about stability results. Okay, and present and provide conclusions regarding storage condition, retest data, or shelf life. It can be provided either in a tabular, graphical, or narrative form. Just a second. So take for example, if you are performing a long-term study, a long-term stability study, then you have specific storage conditions that needs to be followed, and there's a minimum time period covered by data at submission. For example, long-term study data are done for 12 months. Intermediate study data, stability studies are performed for 6 months, and accelerated are also performed for 6 months, but with different storage condition. So all the data that are generated during your stability testing is submitted in 32S7 and summarized as well. Okay. In post approval stability protocol and commitment, you have to include study storage conditions and minimum time period covered by data at submission. Right. So this is about your 32S drug substance. If you have any questions here, any question related to drug substance or any guidelines that you think you need specification for, you can ask them in the chat box, and then we'll move to 32P. Just go through the ICH guideline for 32S once again, and if you feel there are any doubts, you can always ask them. Okay, let's move forward with 32P. See, 32P is for your drug product. 32S was entirely dedicated to drug substance, API, raw material, and 32P is your drug product, which is your finished product. Okay. In 32P, we have 8 sections, 32P1 to 32P8. 32P1 talks about description and composition of the drug product. The information provided should include description of the dosage form. For example, it's a tablet or a capsule or a suspension, solution, water for injection, etc. So that description of the dosage form will be provided in description of the dosage form. Then the composition, you have to list all the components of the dosage form and their amount on a per unit basis including overages if there are any, the function of the component, if it is a, if it is a diluent. Yes, I can definitely send this guideline PDF. I'll share it with the team, and they'll share it with you. Okay, coming back to composition. So you need to list all the components of the dosage form and their amount on a per unit basis. You also have to provide the function of the components. For example, if you're using a diluent, if you're using glycerol, you have to make sure that the function of glycerol is mentioned in under description and composition of the drug product, along with reference to their quality standards. So in USP, BP, compendial monographs or manufacturer specifications can be referred to mention the quality standards in 32P1. Apart from this, description of accompanying reconstituent diluent needs to be submitted, and the type of container and closure used for dosage form and accompanying reconstitution diluent should be explained here. Take for example, you are using an amber colored container for the syrup. You need to mention that amber colored container is being used and the composition of that amber colored container. Okay, what all products were used, etc. Take for example, you're using an IV injection and a reconstituent diluent. So the container and closure for both the IV water for injection and the drug product or will be submitted in description and composition of the drug product. Then is your second part of drug product, which is pharmaceutical development, which is a very important section of the dossier. In pharmaceutical development section, information on the development studies conducted to establish that the dosage form, the formulation, the manufacturing process, container closure system, microbiological attributes, and usage instructions are appropriate for the purpose specified in the application. If you are specifying that the drug product will be used to treat ulcerative colitis, then you need to make sure that all the data that you're submitting is appropriate for the purpose specified and not deviate from that. So the studies described here are distinguished from routine control tests conducted according to specification. Additionally, this section should identify and describe the formulation and process attributes, critical parameters that can influence batch reproducibility, product performance, and drug product quality. Okay, any other supportive data or results from specific studies or published literature can be included within or attached to the pharmaceutical development section for easy reference, and other additional supportive data can be referenced to relevant non-clinical and clinical sections of the application. Like I told you, today we submit DMFs in eCTD format. We submit dossiers in eCTD format. So it's very easy to reference your data to any of the clinical study or non-clinical study reports that you're submitting in that dossier. Okay. So under pharmaceutical development, there are further sections. The first one is your component of the drug product. Okay. Under component of drug product, we talk about drug substance and excipients, both. Because we are formulating or manufacturing the drug product in using both drug substance as well as the excipients to make the finished product. So under drug substance, the compatibility of the drug substance with the excipients listed in 32P1 should be discussed. 32P1, we talked about composition. So all the excipients will be listed here, and when we talk about the compatibility of drug substance with the excipient listed in 32P1, we are talking about the excipients we've listed in 32P1. Okay. So Compatibility needs to be discussed, and additionally, any key physiochemical characteristics like water content, solubility, particle size distribution, polymorphic or solid state form of the drug substance that can influence the performance of the drug product should be discussed. So all the physiochemical characteristics, it's very important to understand the physiochemical characteristics to make sure that the drug product's performance is not hindered by physiochemical characteristics. The water content should fall into the specific limits set for that specific drug product. All the limits are set for the specific drug product, and we need to make sure that they are falling and they are compliant. Okay. For combination product, the compatibility of the drug substance with each other should also be discussed. Okay. Take for example, a drug product contains two different APIs. So their compatibility needs to be studied in order to make sure that the performance of the finished product is not hampered. Then comes your excipient. So the choice of excipients listed in 3 to P1, their concentration, their characteristics that can influence the drug product performance should be discussed relative to their respective functions. Discussed here to be under components of the drug product. Then, once we've established the substance and the drug and the excipients, we will move towards 3 to P22, which will talk about drug product, its formulation development, averages that are present, and the physiochemical and biological. So under formulation and development, a brief summary describes the product into consideration the proposed route of administration and usage. For example, syrup. Syrup is for there are suppositories for uh, citric or in. So there are IV, there are injections which can be intramuscular or intravenous, etc. etc. So the route of administration here as well. And the results from comparative in vitro studies or in vitro studies will be disintegration, dissolution, etc. For example, there is a new drug for which about for generic drug or an ANDA. You need to make sure that the ANDA drug is bioequivalent to the. Then is your overages in the formulations described in 3 to P1 should be justified under 3 to 2. Then 3 to 2 talks about physiochemical and biological properties. So any parameters relevant to the performance of the drug product such as pH, ionic strength, dissolution, redispersion is that you can think of. Or biological to the drug product. 3. Okay, so this also makes it a very important section of the dossier 2P22 and 3 to P21. Then comes your 3 to 3 about manufacturing process development. In substance also we've talked about manufacturing in the manufacturing site or will be discussed here. So the selection and optimization are the critical be explained in manufacturing process development. Where relevant, the method of sterilization should be we talk about be sterilized before administration. But we come under manufacturing process development. Any differences between the manufacturing process used to produce clinical batches and the process described in Then is your section 32P24, which talks about so all the parameters will be discussed and explained in container closure system for the drug product in section 32P24. Then comes your 32P25, which talks about biological attributes. Okay, where appropriate, the testing. Okay, am I audible now? Okay, I'll discuss microbiological attributes again. So, where appropriate, we need to make sure that the microbiological attributes of the dosage form are discussed, including the rationale for not performing microbial limit testings for non-sterile products. For sterile products, you have to perform the microbial limit testing. But for for non-sterile products, if you're not performing microbial limit testing, you have to provide a rationale for why the testing hasn't been performed. Okay, along with selection and effectiveness of the preservative systems in the product containing antimicrobial preservatives. For sterile products, we are using antimicrobial preservatives, so the effectiveness of that preservative system should be discussed and defined here under microbiological attributes. For sterile product, the integrity of the container closure system to prevent microbial contamination should also be discussed. We are using airtight containers to prevent microbial contamination. So all of that should be discussed here under microbiological attributes. And the last is your 32P6, which talks about compatibility. The compatibility of the drug product with both secret solvent.